Swayam Prabha. Digital India. Educated India.
Under paleontological evidences, we will discuss how missing links represent an example in support of evolution. Then we will discuss how the phylogeny of various organisms which has been traced on the basis of fossils can be used as an evidence for evolution. And lastly, we will discuss the example of living fossils. So starting with the what is paleontology? Paleontology is simply the study of fossils. Now what are the fossils? Fossils are any body or traces of a body, animal or vegetation buried and preserved by natural means. So any organisms or its parts which has been preserved by natural means, they are called as fossils. It's Leonardo da Vinci who has been credited as the father of paleontology, whereas George Cuvier is called as father of modern paleontology. What are the missing links? Any specimen or a species of an extinct organism thought to be intermediate between two clades and so to provide a link between them. In simple words, it is a fossil which has been shown to have characters of two different clades, two different taxon, two different species. And thus it provides as a linking evidence. But remember, this is a fossil. The example may include the famous Archaeopteryx, which is a missing link between reptiles and the birds. Yet another example can be Simoria, which is a missing link between amphibian and the reptiles. Let us discuss Archaeopteryx in a bit detail. This is the Berlin specimen of Archaeopteryx and this is the reconstructed view of Hillman of Archaeopteryx. This Archaeopteryx showed reptilian features in terms of teeth and the tail vertebrae, whereas it also showed the avian features including the wishbone and the feathers. So this fossil is supposedly a missing link which used to have a connection between the birds and the reptiles. The Archaeopteryx is considered as a cousin to lineages running from reptiles, particularly the dinosaurs, to birds, as in the words of Thomas Henry Huxley. He says that such predictive forms are predictable consequences of evolution that help to prove the theory. That means such kind of fossil records, such kind of missing links, they are a strong evidence, a physical evidence in support of evolution, that these birds might have evolved from the reptiles. Besides these missing links, the phylogeny or the evolutionary history of a particular organism which has been revealed through various fossil forms, that also present a strong evidence of evolution. For example, the phylogeny of horse. Richard Owen described the earliest known horse fossil, which is also called as Hyracotherium or Eohippus. Othniel C. March and others have presented the various transitional stages between the 60 million year long journey from the Hyracotherium, the first fossil of horse, to the modern horse, that is Equus, based on the fossils. So we have found various fossils which represent the transitional stages between the various forms of horse, and thus they represent the strong evidence of evolution. Now, if you see these evidence in terms of fossils, which are here in the form of bones or the teeth or maybe the skulls, this have revealed the whole phylogeny of horse. That means the transitions from the low to high crowned teeth. The earlier fossils were having low crowned teeth. The evolution from low to high crowned teeth has been proved by these fossils of teeth. Browsers to grazers form, pad-footed to spring-footed, and small to large brain, etc. So all these have been proved on the basis of fossils of bones, teeth, and the skulls, which represent a strong evidence again in the support of evolution. Then there are something called living fossils, an extant taxon, a living taxon that closely resembles organisms otherwise known only from the fossil record, such as the lungfishes, horseshoe crab, ginkgo plant, and so on. If you see, this is a 170 million year old fossil of ginkgo, and you can see the shape of the leaves here, whereas this is the present day ginkgo plant. So this organism, this plant still resembles the organisms which are otherwise known from the fossil record. So what about the evolution? Has evolution not taken place? What is the direction of evolution? These are the things to ponder upon when we are discussing the paleontological evidences in support of evolution. Moving ahead, let's discuss the evidences which are coming from the biogeography. Here we will discuss the phenomena of restricted distribution of organisms. There is something called discontinuous distribution as well, and then the island fauna. Starting with the understanding of what do we mean by the biogeography. Biogeography is the study of distribution of living organisms on Earth with respect to space and time. Now, the whole Earth, from the point of living organisms, particularly the animals, has been divided into 6 realms, which are called as Palearctic, Nearctic, Neotropical, Ethiopian, Oriental, and Australian. And if you see in current world map, these zoogeographic realms are situated like this. So this is the Palearctic, Nearctic, Neotropical, Ethiopian, Oriental, and then the Australian regions. So what do we mean by the restricted distribution? If you take the example of monotremes, monotremes are the egg-laying mammals just like duck-billed platypus, echidna, etc. They are found only in the Australian region. Australian region is somewhere here. Similarly, if you talk about marsupials or the pouched mammals just like kangaroo, they are found only in Australia, New Zealand, and the South American region. So the question is, why this restricted distribution is there? Why monotremes or egg-laying animals are here only? Why these marsupials are in these restricted pouches in the world and not in the rest of the world? Similar kind of questions do arise when we discuss the phenomena of discontinuous distribution. Now, discontinuous distribution is that the populations of related organisms found in widely separated parts of the world. For example, if you talk about the lungfishes, there are 3 known forms or species of lung species. Lepidosaurin is one type which is found only in the South America, that means this area. Neoceratodus, that is another type, it is present only in the Australia. Protopterus is only found in Africa. So the question is, why 3 forms of lung species are found in 3 separated, so widely separated areas of the world? Probable answer is that these 3 parts probably were initially part of the same mainland, and because of their disconnection, because of these oceans, these three forms got separated and then started evolving in different areas, like South America for Lepidosiren, Neoceratodus in Australia, and Protopterus in Africa. Same is the case with other examples like camels, which is found in Asia, and their closely associated member, that is llamas, which are present in the South America. So again, probably initially they were part of the same mainland, but with the evolution of Earth, with the geographic changes, they got separated and thus got evolved separately in different geographical regions. So this biogeography again represents an example of evolution or evolutionary evidence. Third type which we can discuss under biogeographical evidence is coming from island fauna, and we can take an example of Darwin's finches, which has been closely and very elaborately studied by David Leck. You might be aware of different forms of Darwin's finches which he explored on Galapagos Island. Leck said that they have evolved from a common ancestor which was seed eating and a ground finch, from which all the different forms, which might be the insect eating, cactus seed and parts eating, seed eating, and birds and fruit eating forms have developed. So different beak types and the size of these finches have evolved with time from this common ancestor. So this is a very prominent example of evolution taking place in nature. Moving ahead from biogeographical evidence, let us discuss about taxonomical evidences. In taxonomical evidences, we will discuss 2 points, that is taxonomy, that is classification, and how the connecting links are presenting an evidence for evolution. Taxonomy is the branch of science which deals with naming, defining, and classifying the organisms in the different groups based on the shared characteristics. Now, this grading of animals in different subsequent groups of complexity, probably or particularly the increasing complexity, presents an evidence of evolution. The natural system of classification as by Carolus Linnaeus is based on similarities which arise due to common ancestry. So again, there is the phenomena of common ancestry which has led to evolution of different life forms. So classification in itself is dependent on similar characteristics which do arise because of common ancestry. You might also have come across the connecting links, which are again the evidence of evolution. Just like missing links were the fossil forms, the connecting links can be the living forms as well. Peripatus, for example, is the connecting link between annelids and arthropods. It shows arthropodal characters like jointed paired legs, compound eyes, and the tracheae. It also had annelidan characters. It has a worm-like segmented body, which is non-cutinous cuticle bearing and has the segmental nephridia. So this organism shows characters of both. So it is a sort of transitional stage. But this is living in contrast to what we discussed about the missing links. Peripatus is not the only example of connecting links. We have number of examples like Euglena is a connecting link between plants and animals, Neopilina between annelids and mollusks, Chimera between a cartilaginous and bony fish, and many such type of examples are there. So again, this connecting links is a strong evidence that evolution has taken place in nature. Then let us move to cytological evidences, or the supporting evidence which are coming from the cell biology studies. Under this, we will discuss how the cellular structures and the structures of its organelles, the protoplasm, chromatin and genetic code represent an evidence of evolution. Starting with the cell and its organelles. If you compare the structure of cells across different organisms from belonging to different species or different genera and so on, except for a minor differences, the basic structure and the organization of all the cell types is almost the same. And not just the cells, but the types, the structure and the functions of various organelles which are present within the cells, just like mitochondria, Golgi bodies, endoplasmic reticulum, etc. They are also same in all these organisms. So this is a strong evidence that they do have still the same basic pattern, and hence they might have evolved from this basic pattern as well. Similar to cell and organelles, if you compare the protoplasm, the chemical composition of protoplasm in all the cells, irrespective of which class or organism they belong to, it's similar. Chromatin, which is made up of nucleic acids and proteins, it is also same across the species or organisms. The composition of DNA is also the same, which comprises of the nitrogenous bases, which might be the ATGC bases, the phosphoric acid and the deoxyribose pentose sugar. Besides this, you might have come across the universality or the universal nature of genetic code, which comprises of triplet codon. So this is also same across all the organisms and species. These are the cytological evidences which indicate that all the life forms might have originated from same basic patterns, from the same origin and same process. Moving ahead, let's go a further level down to the molecular level. And in molecular evidences, we will discuss about the phylogeny which is operating at the molecular level. That means evolution occurring at the molecular level. And then we will slightly touch upon the sequential homology. That means homology which is existing in the sequences of biomolecules like proteins and the nucleic acids, which comprises of orthologous, paralogous and xenologous sequences, which we slightly discussed in homology. So, in phylogenetic relationships, when we discuss these phylogenetic relationships based on the molecular sequences, just like amino acids in case of proteins or the nitrogenous bases in case of nucleic acids, using various bioinformatics approach, this comes under the molecular phylogeny or molecular evidence of evolution. Now, for example, this is the sequence. This is an amino acid sequence of a particular protein, say histone H1, which has been taken from different organisms, let's say human, chimpanzee, mouse, rat and cow. Now, if you compare their sequence, wherever you see these stars, colon, etc., these symbols represent whether this particular amino acid has been conserved across the organisms or it has gone under some change. So the star indicates it has never undergone any change. K is the lysine. So at this position in different organisms, histone H1 has all the lysines, whereas if you compare this, there might be a change occurring at different organisms. So this kind of bioinformatic approach again provides an evidence that how the basic structure or the common sequence has been there in different organisms of the conserved proteins. Now you remember this figure we discussed earlier, where an ancestral X gene has undergone gene duplication and given rise to different forms X.1 and X.2, which because of speciation has gone to humans and chimpanzees. So this human X.1 when compared to human X.2, that means the genes or sequences which are arising because of duplication, they are called as paralogs. Whereas the sequences which are arising because of speciation, let's say human X.1 compared with chimps X.1, they are called as orthologs. And when these are compared with the bacterial protein, far distant protein, they are called as analogous ones. There is another category which is called as xenologous. Now in this xenologous, let's say there is a gene A which has undergone to species 1 and species 2 because of the speciation process. Now the part of it, or let's say gene A2, because of HGT refers to horizontal gene transfer, has gone to this particular organisms of species 1. So species 1 has A1 and gene A2, and this gene A1 and A2 are xenologs of each other. So orthologs do arise because of speciation, paralogs arise because of duplication, and xenologs arise because of the horizontal gene transfer or the lateral transfers. So these are again the molecular evidences of evolution or homology in sequences at the gene level or protein level which support the evolution theory. Let us move to last series of our evidence which come from the fields of physiology and biochemistry, under which we will discuss the various enzymes and the metabolic processes, the hormones, the conserved proteins like cytochrome C, the body fluids which may comprise of blood and lymph, blood proteins, blood groups, and various excretory substances. They also represent an example of evolutionary evidence. Let us see how. Starting with enzymes or metabolic processes. There is a similarity in organic substances and the biochemical pathways or process like the anabolic and the catabolic pathways which are occurring in the cells, the protein synthesis pathways in different organisms. So this similarity of organic substances and biochemical pathways which is existing in different organisms, it indicates that there has been a common origin. Various steps in biochemical pathways, if you compare, let's say the glycolysis pathways, all these steps are conserved across the organisms. And not just the pathways or the steps, but the enzymes which are catalyzing these steps, they are also same. The digestive enzyme, for example, if you take an example, in all the organisms or animals, like the example of trypsin or amylase if you take, they do have similar kind of structure and functions. So all these similarities provide a hint that there is a common origin of all these forms. Not just enzymes, if you consider the hormones, they are also having similar structure and functions in different organisms. For example, you can take the example of thyroid hormone or insulin. In different organisms, they are structurally so same, they are functionally so related that they can be transferred from one organism to another organism belonging to different species. For example, pig insulin can be taken and given to human insulin, human forms. So their structural and functional similarity is again indicating the common origin of different life forms. Same is the case with cytochrome C, which is a respiratory pigment of eukaryotic cell. It has identical or very similar composition across all the organisms which we have come across. If you take the example of blood and lymph, that is the body fluids or the blood proteins or the blood groups, there is a similarity in terms of composition and physiological role of these fluids in all the vertebrates. The similarity and differences in blood groups, let's say ABO blood group, is also an indicator of evolution. And the evolutionary relationships based on the similarity and dissimilarity of blood proteins in different species can be traced. So based on the proteins which are found in blood, based on the blood grouping, we can also trace the evolutionary history of different species or different organisms. You might have made hemine and hemochromogen crystals. So you can observe how similar these structures are within a species. What is the dissimilarity, if it is, across different organisms, across species? These are the indicators of evolution and hence a strong evidence in case of evolution. So let's quickly discuss what we have so far learned in this lecture in the form of summary. We discussed various evidences of evolution. So from the fields of morphology and anatomy, we discussed the structures which are called as homologous, analogous, the vestigial organs, parallel structures, and the atavism phenomena. Then in case of embryological evidence, we discussed about the ontogeny and the recapitulation theory or biogenetic laws, which is an evidence from the fields of embryology for the evolution. Then we move to fossils as paleontological evidence of evolution in the form of missing links, the phylogeny history of particular organisms, where we took the example of horse phylogeny, and the living organisms, living fossils, sorry. Then we went ahead with the biogeographical evidences of evolution, where we discussed the phenomena of restricted distribution, discontinuous distribution, and island phenomena in the form of Darwin's finches. Taxonomical evidences come from the classification and the connecting link examples. Then the cytological evidence from the similarities in the structure and function of cell, its organelles, protoplasm, chromatin, and genetic code that we discussed. At the molecular level, we discussed the sequence homology in terms of orthologous, paralogous, and xenologous sequences, and how the bioinformatic approaches have proved the existence of evolution. And lastly, we discussed various physiological and biochemical pathways, molecules, enzymes, hormones, proteins, etc. How these represent the evidence of evolution. So here we'll end this lecture with these exhaustive discussion of evidence of evolution. For more discussions, you can go through these books on evolution by Ridley, Hall and Hallgrimson, and by Douglas Futuyama to understand different domains of evidences to support the theory of evolution. With this, I would like to thank you. Happy learning.